Greetings everyone! Today we will explore how cells change over time with single-cell RNA-seq. We will use Scvelo to calculate the RNA velocity, showing the direction each cell is moving, and CellRank to predict their final fates. You don't need any external dataset. We will use built-in example data, so you can follow along. Without further ado, let's get started. To start, we need a dataset that already includes information required to study RNA velocity, that is how genes are being turned on or off over time inside each cell. Each cell in our dataset should have two types of RNA measurements, either spliced RNA, which is mature RNA molecules that are ready to be used by the cell, or unspliced RNA, which is newly made RNA molecules that haven't been processed yet. And by comparing the amount of spliced versus unspliced RNA, we can estimate whether a gene is being activated or repressed, which helps us predict the cell's future state. So let's load our libraries. Then let's make some settings for nicer plots. Now let's load a built-in example dataset. We can choose a data from pancreas. And now let's print a summary. Let's run our code. And here we have it. As you can see, we have here 3,696 times 27,998. This means that we have 3,096 cells and 27,998 genes, proper spliced and unspliced layers, that's needed for RNA velocity. So as you can see, this table is part of our annotation data object's metadata. It describes each cell in our single-cell dataset. Each row is one cell, and each column gives the information about that cell. And as you can see, we have different columns. Index, which means the unique cell ID barcode, like for example, AAA, CC, TGAGA, and so on. This identifies one single cell. And cluster scores, this means broad cluster label, general cell type group, like pre-endocrine, ductal, endocrine, etc. And clusters, more detailed or refined cluster label. For example, endocrine, you might see specific types like alpha or beta. S score, a numerical score showing how active S phase genes are, like DNA replication part of the cell cycle. For example, negative value, not in S phase. Positive, actively replicating DNA. G2M score, a numerical score showing how active G2M phase genes are, mitosis part of cell cycle. Negative, resting cell, but positive, dividing cell, and so on. Now let's perform step 2, which is preprocessing and normalization. Before computing RNA velocity, we need to filter low-quality genes, those that are not detected often, and normalize counts so all cells are comparable. So Let's run our code. And here we have it. So as you can see, we filtered out 20,801 genes that are detected 20 counts. So that means out of 27,998 total genes, about 20,801 were low quality or rarely expressed. So only about 7,000 remain, and later only the top 2,000 are used for velocity. This is normal, it removes the noise. And for normalized count data, X spliced and unspliced as we see. So normalization makes sure all cells are comparable, so velocity estimation doesn't get biased by high count cells. So let's go for step 3. We estimate for each cell and gene how the expression level is changing. That is whether it's increasing upregulated or decreasing downregulated. It's done by comparing unspliced versus spliced mRNA counts. So unspliced, the newly made RNA, shows what's about to increase, and for spliced, mature RNA shows current expression. From that, we infer velocity vectors, showing the direction of gene expression change. So, we write here stochastic, as stochastic is robust and works well for most datasets. RNA velocity for each cell, that is how its gene expression changes over time. So this code estimates each cell's transcriptional speed and direction. Let's run our code. So this code computed the RNA velocity vectors for each cell, basically arrows that show how each cell's gene expression is changing. So now our data has a data layer's velocity. The per gene velocity estimates either positive for the expression increasing or negative, which is decreasing. And now let's go for step 4 to visualize our RNA velocity. Let's run our code. And here we have it. So as you can see, each point is a single cell, and the cells that are transcriptionally similar are closer together. Colors means different cell clusters, like cell types or states, and the arrows or streamlines equal to RNA velocity predicted prediction of a change, how cells are transitioning from one state to another. So if arrows are flowing from one cluster to another, that suggests cell differentiation or fate transition between those clusters. So in our dataset, which is pancreas development data, the main developmental process is endocrine differentiation, how precursor cells become hormone producing cells like alpha, beta, and delta. So, for example, here, arrows from pre-endocrine to the beta that shows that some cells are moving along a developmental path toward the beta cell insulin producing identity, and the arrows from pre-endocrine to alpha that indicate another group is differentiating into alpha cells like glucagon producing. So together these arrows reveal branching developmental trajectories meaning same precursor population, which is pre-endocrine, splits into multiple mature endocrine fates. And regarding the biological story behind this plot, for example here, ductal cells. These are the starting progenitor cells, the eldest stage in the lineage. And then NGN3 low EP here to NGN3 high EP. It's the pre-endocrine path. Cells progressively activate endocrine differentiation programs. The arrows flow forward, showing the transitional route toward the endocrine identity. And then the endocrine branches, which are beta, alpha, and delta. And now let's start step 5, which is identify terminal states with CellRank. But why we use CellRank? From the previous step, we saw directional arrows tells us which way cells are moving, for example from pre-endocrine to beta, but velocity alone is local. It shows direction near each cell. It doesn't tell us where the whole population ends up, where it starts, or what the probability of each fate is. That's where CellRank comes in. CellRank takes the velocity graph and converts it into a Markov model of cell transitions. Then it finds macrostates, broad stable groups of cells that behave similarly, like clusters, but learned from the dynamics. It identified terminal states, for example, alpha cells or beta cells. And it identified initial states, early progenitors with mostly outgoing transitions. In addition, it computes fate probabilities. For example, this cell has 0.8 probability of becoming beta cell. So what is a kernel in CellRank? In CellRank, a kernel is a mathematical object, a matrix, that says given cell A, what is the probability it will be transitioned to cell B. And we have two main kernels, velocity kernel and connectivity kernel. Velocity kernel built from RNA velocity directions, and connectivity kernel built from the neighborhood graph. It captures structural similarity. For example, cell A and cell B are transcriptionally similar. But why we combine them? If you only use the velocity kernel, the model might be noisy or biased, since velocity estimation is not perfect. If you only use connectivity, you lose direction. So CellRank mixes them, usually 80% velocity and 20% connectivity. So let's build and combine our kernels. And now let's combine them. Let's run our code. Now let's print them. As you can see, velocity kernel and connectivity kernel. Now we want to find groups of cells that represent major fates. For example, these cells will become alpha cells, these will become beta cells, and so on. So And now let's plot our macrostates on the UMap. We choose all. Let's run our code. And here we have it. As you can see, we made a map of our cells with the UMap that shows how cells are grouped based on how they behave over time. We have three different colors: blue, orange, and green. For example, each is a macrostate, which means a big cluster of cells that share a similar fate or behavior pattern. You can think of it like each point is a single cell, each color a group of cells traveling in a similar biological direction, and the chart shows where cells are moving in their developmental journey. So right now, our map shows the main fates 0, here, 1, and 2. But it doesn't yet know which groups are starting and which are ending. And that's what we are going to do in the next step. So let's run our code. Now, once we ran those lines, CellRank will mark which parts of our new map are starting and which are ending. After that, we can calculate how likely each cell is to go toward each fate. That is a fate possibility. And here we have it. As you can see, we have three fates. Fate probability is 0, and for 1 and for 2. Each cell in the plot has a fate. That's the model confidence that the cell will end up in a particular terminal fate. And we have three colors. Blue is from 0 to zero point 0.2. The cell is very unlikely to become this fate. It's far away in trajectory space. And the green color, which is about zero point 0.4 to zero point 0.6. The cell is intermediate, maybe still transitioning. And the yellow one, which is the cell is very likely to become this fate. It's near the terminal state. So, for example, yellow cells are those that are almost mature here, or fully committed to fate 0. And the blue ones, they might be heading toward fates 1 or 2 instead. So cells on the upper right are bright yellow, meaning these are most likely to differentiate into fate 0. And the rest, purple, will not go toward this trajectory. This region represents one lineage or branch of the differentiation process. And regarding fate 1, most of the UMAP is brightly yellow, so many cells are predicted to go toward fate 1. This likely represents the dominant or default differentiation path in our dataset. These cells are biased toward the same terminal state, major mature cell type. And for fate 2, only a small subset, which is in the top left corner here, the yellow one, meaning that only few cells are committed to fate 2. It's probably a minor lineage or rare terminal state. So fate 0 alpha The yellow high probability region is concentrated on the alpha cluster. Those cells are strongly predicted to become alpha cells. Surrounding areas are greenish, moderate probability, meaning some nearby cells are partially biased toward alpha, but not fully committed. And fate 1, beta, the bright yellow area, sits on the beta cluster. This is the main beta cell differentiation path. Other regions are greenish yellow, about moderate potential, and fate 2, ductal, the yellow spot is over the ductal region. Cells there tend to remain or revert to progenital identifier rather than differentiate.
And here we've reached the end of our analysis. If you'd like us to make a tutorial or episode about another topic, feel free to leave your suggestion in the comments below. Don't forget to like this video. Thanks for watching. See you next time.